Okay, so let's work our way through the study guide. Again, this study guide is really just a little part of what you need to know for the exam. This is mostly just stuff from the first part of the chapter, and then at the end there's some kind of patchy stuff from the second and third lectures. But then you have the chart, and the chart is where all the meat is, like for the actual hormones, where they come from, where they go, what they do. So let's work through this first. This form of intercellular communication utilizes chemical messengers that go into the bloodstream. Endocrine, right? Hopefully you got that, because that's the whole point of all of this. This form of intercellular communication uses chemical messengers that go through the extracellular fluid. Paracrine. What form of intercellular communication is used by cardiac muscle cells? It's where information is transferred from one cell to a neighboring cell. Direct. What's the other example that we talked about that uses this? Cilia movement. So coordination of cilia movement, like for example in the respiratory tract. Remember, like the trachea, the bronchi, the cilia all move together to push the mucus. It wouldn't make sense if they were going like all crazy against each other. All right, they've got to go together. So they use direct communication to control that. What form of intercellular communication uses neurotransmitters that go across the synaptic cleft? Synaptic, right? Across the synaptic cleft. Endocrine glands versus exocrine glands. So endocrine glands secrete hormones into the bloodstream, and do they have intracellular or extracellular effects? Intra. Right? They bind to receptors and they cause some sort of effect in that cell. Exocrine glands release secretion via ducts onto an epithelial surface, right? So it's like going onto your skin, like sweat glands, or going into your stomach, some sort of a surface. And they have, are the effects intracellular or extra? Extra. Good. They have extracellular effects, right? Like when sweat evaporates, you're not changing anything inside a cell. I'm cooling my body off. Which regulatory system do we utilize to handle urgent stresses when we need a really quick response? Nervous system. The nervous system, right? The endocrine system takes a while. So if I'm falling down and I need to move my muscles to catch myself, I'm not going to rely on the the endocrine system to do that. The nervous system handles that because it's fast. So I can rate myself very quickly, right? Or senses. If I'm seeing, if I'm hearing, I need that information to travel quickly, so I'm going to use the nervous system. Which regulatory system do we utilize to control growth, development, reproduction, and metabolism? Endocrine, right? Those are slower processes. It doesn't matter if it takes a few minutes or a day or whatever to make it happen. They're slow, ongoing processes. When the endocrine system is used to regulate the body, it results in what kind of effect? So think timing, like how long it lasts, how quick, whatever, and then the targets, if they're widespread or narrow. So endocrine system results in a widespread effect, meaning if I release a hormone here, I can affect cells all over my body, right? It's not just right there that I'm affecting. I can affect everywhere. A widespread effect, and then we said long-lasting, right? Endocrine communication results in, so random, what? Especially compared to synaptic communication. Again, think timing and targets. Yeah, I mean, this is kind of, this is like the same thing, sorry, pardon French. Yeah, this is like the same thing. It's a widespread effect, right? It's a long-lasting, so it's used to kind of maintain long-term effects. So not anything you need a rapid response for, something that's ongoing and longer. Again, like growth, reproduction. I don't know why I put that. It's not the same idea. Which class of hormones includes small molecules synthesized from the amino acid tryptophan or tyrosine? So what are those called? Amino acid derivatives. derivatives. Give me 2 examples of amino acid derivatives.
Melatonin and thyroid hormones.
Okay, thyroid hormones, which come from which amino acid? Tyrosine. And what was the other one?
Melatonin.
Which comes from what? Tryptophan. And do these bind intracellularly or extracellularly? So thyroid hormones, where do they bind? Intra. Thyroid hormones are the weird ones. They enter cells and bind intracellularly. Where does melatonin bind? Extracellularly. Okay. We also did like epi and norepi. Those are bind extracellularly as well. Steroid hormones are examples of what class of hormones? Lipid derivatives, right? Steroids are lipids. They're made out of cholesterols and fatty acids, or fats. So give me some examples of steroids. Androgens, like what? Like testosterone. Okay, what else? Estrogen, or estrogens, there's a lot of them. What else? Progesterone. What else? Corticosteroids, like if I said cortisol, right? I could say cortisol. We talked about hydrocortisone. Aldosterone. Right, there's lots of them. If it ends in "on," progesterone, testosterone, aldosterone, you can bet that it's probably a steroid. What class of hormones consists of chains of amino acids?
Peptide hormones.
Peptides. Most of our hormones are peptide hormones. Do these bind intracellularly or extracellularly?
Extracellularly.
Extra. Remember, they're very big and they're not lipid soluble, so they cannot enter the cell. They have to bind outside of the cell. What does that mean? What do they have to utilize?
Second message.
Beautiful, second messengers to go exert the effects inside. Which hormones bind to intracellular receptors? The lipids, right? So like understand that I could, like when you're going through this, think about all the different ways I could ask this. So I could give you like A, B, C, D, and E, and I could say which of the following does not bind intracellularly. So I'm not gonna put just lipids. I could put lipid derivatives, I could put steroids, I could put aldosterone, I could put progesterone, I could put estrogen, and then what else besides lipids? What's the weird one? Thyroid hormones. Okay, so like, I don't know how to get rid of that. Go away. Okay. So like when you write something like lipid derivatives, understand what that means. Like what it is that you're actually saying by saying lipid derivatives. Okay. Why do some hormones travel in the bloodstream bound to a carrier protein? To give them solubility in the blood. Right? So some things, remember, don't like water. They're hydrophobic. They do not like water. So we got to add a protein onto them in order to make them soluble in the bloodstream so that they don't clump, so that they'll spread out nicely in the bloodstream. What does this do to their clearance?
What do you mean by clearance? Just like the length that they exist in the body?
Good. It prolongs or decreases their clearance. It lengthens the amount of time that it takes to clear them so they remain in the body for a long period of time. Remember, the kidneys won't filter them out when they're bound. The kidneys filter based on size. So when we add a huge carrier protein, they're big and they don't get filtered out of the kidneys. And then what did you say this does to their time of effectiveness? Increases the time of effectiveness. Okay, so remember we talked about like thyroid hormones, and we said you could take that thyroid gland out and you still have a week worth of thyroid hormones because they're bound. Okay, know that this statement right here, the presence of a hormone receptor determines sensitivity to a hormone. Right? The concentration of the hormone does not determine sensitivity. The concentration of the hormone can change the number of receptors you have so it can influence sensitivity, right? But the presence of a hormone receptor determines sensitivity to a hormone. What I mean by that is if I have a cell here, if that cell does not have any receptors for hormone X, it doesn't matter, it's not sensitive to hormone X. It is not sensitive to X. It doesn't matter if I have a really big concentration of hormone X, it's simply not sensitive to it because it does not have receptors. Okay, it's the receptor that determines sensitivity to a hormone. We'll talk about how we can change the number of receptors right now. So this is what I mean by the concentration of the hormone can affect the sensitivity, right? Because we can change the number of receptors we have based on concentration, but it's actually the presence or absence of the receptor that determines the sensitivity. Half the class gets that wrong every time, so make sure you get it. So when would hormone receptors be down-regulated? So I have a cell here, and this cell has this many receptors. What would cause this cell to down-regulate, to decrease the number of receptors it has? High, good job, high hormone concentration. High hormone concentration. All right, if I've got a ton of this hormone here so that I'm just constantly stimulating it, the cell's gonna say too much. Too much. I'm gonna decrease the number of receptors that I have so I'm not being stimulated so much. Down-regulate its receptors. So when you have a ton of the hormone present, you down-regulate your receptors. Does down-regulating make you more sensitive or less sensitive? Less. Makes you less sensitive. Okay, so what about upregulating? If I have my cell, and my cell looks like this, and I barely have any hormone, right? The hormone's few and far between. The cell might say, I'm not getting stimulated enough. I'm gonna upregulate my receptors, right? I'm gonna add another receptor over here so that I'm more likely to get more of a signal, right? So you add more receptors. So when would you do that? You would add more receptors when hormone concentrations or levels are low, you upregulate, you get more receptors. What do we call that as far as sensitivity? If I'm having more receptors, am I more sensitive or less? More. I'm more sensitive. What determines sensitivity to a hormone? Receptors. Yes.
Isn't it like a mutual relationship?
One affects the next, which affects the next, but the concentration is not directly correlated with sensitivity because this cell could have.
Right, but the concentration is directly correlated to receptors.
But not always. This cell might not have receptors. It might not be sensitive to that hormone. And it doesn't matter what I do to the concentration. It's never going to be because it's just not supposed to be sensitive to that hormone. Okay. So like estrogen does not affect every cell in your body. So it doesn't matter what the concentration is. The concentration could go anywhere and some cells will just simply not be sensitive to it. Why? Because they don't have receptors. So ultimately it's the receptor that determines sensitivity. Concentration can affect the number of receptors, but it's not across the board. Not everything gets upregulated and downregulated like this. Some things don't. And some just won't have a receptor, period. Is that okay? I know that's kind of a weird concept.
It just, I feel like it was worded differently in the PowerPoint.
The PowerPoints go through it just like this. Like they say receptor sensitivity, or they say receptors determine sensitivity. Like the presence or absence of a hormone receptor determines if it's sensitive. And then it goes through and describes what upregulation is and downregulation. And I get that seems confusing because yeah, like the concentration of the hormone affects the number of receptors, which then correlates to the sensitivity. Okay. So it can affect it. But if you're talking about what flat out determines sensitivity, it is receptors. Okay. If you go back and look at it and have a question, we can talk about that. Sure. So, like, and maybe it's this type of a sentence that's confusing. So when do cells become more sensitive to a hormone? When levels are low, they'll become more sensitive. How do they become more sensitive though? upregulation, they get more receptors because it's the receptors that determines how sensitive they are.
So cancer is upregulation?
No. They become more sensitive when hormone levels are low. How do they do it? Upregulation. When do they become less sensitive? When levels are high. And how do they become less sensitive? Downregulation. Good. The lipid derivative class of hormones share what important structural feature? 4 carbon rings. Right? Those really non-polar carbon rings that make them lipids. In general, which hormones would be the longest acting? Okay, so in this, like what I'm referring to are how binding proteins will affect this length and which ones are bound. Okay, so which kind of general types of hormones would be the longest acting? Steroids or lipid derivatives and thyroid. Why?
Because they're not soluble in blood.
Good, so that means they have a what?
Carrier.
They have a carrier protein binding. They've got the protein attached. Good, perfect. Again, same thing for that question. Know what you're saying when you say steroids or lipids. I understand what hormones you're talking about there. Hormones that bind extracellularly have to utilize what? Second messengers. Remember, the hormones that bind messengers, that bind extracellularly, never actually enter the cell themselves. So they have to have something else kind of go inside and do their work for them. That's a second messenger. CAMP or cyclic AMP is one of the most common second messengers. We also utilize cyclic GMP. We utilize calcium ions frequently. The release of hormones from the anterior pituitary is controlled by what? The hypothalamus. So what comes from the hypothalamus? Releasing and inhibiting hormones from the hypothalamus. Know what each of the regulatory hormones from the hypothalamus does. That's straight from the chart, right? So we'll kind of talk about that if we have time to talk about the chart. But it's super, super self-explanatory, right? If it says releasing factor, it causes the release of whatever it is it's talking about. If it says inhibiting factor, it inhibits the release of whatever it is that it's talking about. It's a pretty straightforward. The only time when that might be hard is if it's like corticotropin. You have to know that that's associated with adrenocorticotropic. But it's pretty self-explanatory. Note the difference between the anterior pituitary and the posterior pituitary in terms of the hormones released and the way that they're produced released. So what I mean by this is that, so know which hormones come from the anterior and which hormones come from the posterior. And then remember the anterior pituitary we just said is controlled with the by the releasing and inhibiting hormones from the hypothalamus. And how do those releasing and inhibiting hormones get there? The hypophyseal portal system. Hypophyseal portal system. So releasing and inhibiting hormones go through the hypophyseal portal system and go to the anterior pituitary. And then the anterior pituitary makes its own hormones, right? It makes whatever new hormone it's supposed to make. The posterior pituitary, remember, weird. It doesn't actually make anything. Okay, what hormones come from the posterior pituitary? ADX. I'm thinking oxy. Okay, oxytocin and what else? ADH. Oxytocin and ADH. So these hormones, ADH and oxytocin, they're actually produced where? They're produced in the hypothalamus. They are not produced in the posterior pituitary. Posterior pituitary does not produce any hormones. Okay, so those are produced in the hypothalamus, and then remember they just follow their axons down to the posterior pituitary The posterior pituitary just releases them. Okay, just allows them to enter the bloodstream. They're not actually made there. And so that means that releasing and inhibiting hormones do not go to the posterior pituitary, right? The hypophyseal portal system does not go to the posterior pituitary. That's all for the anterior pituitary only. Which of the following correctly describes the release of hormones? Okay, so there will be a question that's what it is. Which of the following correctly describes the release of hormones? And it'll just show you numerous kind of successions, and you have to say if it's correct or not. So you should understand all of the successions of hormone release. So this, for example, TRH, Thyrotropin-Releasing Hormone, leads to the release of TSH, Thyroid Stimulating Hormone. That leads to the release of T3 and T4. Is that correct? Yeah, right? We go from the hypothalamus to the anterior pituitary to the thyroid gland. That's a correct succession of hormones. Growth hormone releasing hormone from the hypothalamus to the anterior pituitary where we release growth hormone. That goes to the liver and we release somatometins. Does that sound correct? Good. The other kind of long stream one that we talk about would be Corticotropin-Releasing Hormone, right? CRH from hypothalamus causes the release of what? ACTH, adrenocorticotropic hormone from the anterior pituitary, and then that causes the release of glucocorticoids, and really also androgens. We don't talk about them much because they're not as significant, but and androgens from the adrenal cortex. Those are the only long things really. The sex hormones, but I don't put those in there. Okay, production of thyroid hormones, T3 and T4, requires what ion? Iodine. What protein? Thyroglobulin. And what hormone? TSH. Over TSH stimulates the whole thing. Yes.
Sorry, can we go back really quick
Yep.
to glucocorticoids, and you mentioned that they could also technically release androgen. Would that also then include the glomerulosa for aldosterone too, because it's the whole cortex?
No, the different segments are controlled differently. The mineralocorticoids are directly released because of the actual concentrations. Those cells have different receptors that respond differently. Okay.